High in the tropical forest canopy, ants move blindly through leaf litter. A single spore of Ophiocordyceps unilateralis lands on the exoskeleton, adhering with microscopic precision. Specialized enzymes degrade the cuticle, allowing fungal hyphae to infiltrate the hemolymph silently. Neuroactive fungal metabolites reach the ant's central nervous system, subtly modulating neurotransmitters like dopamine and octopamine. The ant begins to wander off course, leaving the colony, a behavior hijacked at the molecular level. Muscle and nerve tissues are gradually colonized. The fungus secretes compounds that induce the fatal death grip on leaf veins or twigs. Once positioned, the fungus accelerates its stroma formation, bursting through the exoskeleton in anatomically precise structures. Millions of spores are explosively released, targeting the next generation of ants, continuing the cycle of parasitic mind control. Each step is a precise orchestration of biochemistry and evolution, behavior rewritten, body repurposed, life exploited with molecular perfection. Other insects nearby detect subtle chemical cues, avoiding or investigating. The forest becomes a silent stage for evolutionary warfare.
Inside the ant, fungal hyphae penetrate muscle fibers with microscopic precision. Secreted neurotoxins bind octopamine and dopamine receptors, subtly rewriting locomotor circuits. The ant's normal foraging behavior is overridden by molecular instructions. Fungal compounds modulate gene expression in neurons, ensuring directional climbing. Muscles contract involuntarily, synchronized to the fungal timetable. Hyphal growth continues, secreting enzymes that prevent immune detection and maintain neural control. The ant's mandibles lock in a leaf vein, the infamous death grip, controlled at a molecular scale. Nearby insects react to pheromones and chemical trails, navigating this parasitic manipulation. Every movement is orchestrated, a literal biological puppet show dictated by evolutionarily honed molecules. Eventually, stroma begins to emerge, poised for explosive spore release. The fungus has rewritten behavior, commandeered physiology, and ensured its reproductive success.
Once the ant's death grip secures it, fungal stroma erupts with explosive precision. Millions of spores are launched into the surrounding forest canopy. Air currents, humidity gradients, and leaf architecture guide their trajectory. Each spore carries genetically optimized enzymes for adhesion and cuticle penetration. Nearby ants contact spores, unknowingly becoming the next hosts. The fungus exploits behavior and environmental physics to maximize infection efficiency. Secondary insects, nematodes, and microfauna react subtly to pheromonal cues. This is natural selection at the molecular scale. Targeted dispersal ensures survival and propagation. The stroma continues to grow, secreting light to attract or deter organisms strategically. Every microscopic interaction, chemical, mechanical, behavioral, is evolutionarily refined. The forest becomes a living laboratory of parasitic ingenuity.
Across tropical forests, fungi have independently evolved identical molecular strategies. Octopamine manipulation, enzyme secretion, gene regulation, different species, same biological goals. The biochemical tools are convergent evolution in action, refined over millennia. Fungi detect host physiology, timing infection with circadian precision. Hyphae secrete neuroactive metabolites, targeting specific neurons to modify behavior. Muscle fibers, joint motors, even mandible locking are molecularly orchestrated. Nearby insects' chemosensory neurons respond to subtle fungal pheromones. Every interaction is a dance of molecules, evolution ensuring survival and propagation. These parasites are engineers of behavior, sculpting living creatures for reproductive success. The forest floor becomes a network of biochemical communication and predation. Behavioral control is not the final objective. The host's death is precisely timed. After the ant locks its mandibles, neural control collapses. Muscle fibers rupture. Internal organs are digested in situ. The fungus consumes the host from the inside. Hyphae invade every tissue. The brain is no longer functional. Now reproduction begins. A fruiting body erupts from the ant's head. This structure is not random. It grows at a specific height, angle, and humidity. Spore dispersal is optimized by airflow. Each spore carries genetic instructions for a new invasion. The forest becomes a three-dimensional infection map. Entire ant colonies are reshaped. This is not parasitism alone. It is ecological engineering. Evolution did not create monsters. It created efficiency.